So friends, now I will discuss the last two classes of plant hormones, that is strigolactones and salicylic acid. Now strigolactones are considered to be the carotenoid derivatives, and in 1966 first strigolactone, which was named as Strigol, was isolated from a parasitic weed which is known as Striga lutea, and now it is known by the name of Striga asiatica. Now the name strigolactone was given because first reason was because it was isolated from the species known belonging to genus Striga, and second, upon the structural elucidation of strigol, it was found that they possess a lactone ring in their structure, and therefore the name strigolactone was given, strigo from striga, and the lactone term was given because of the presence of the lactone ring in the structure. Now if we look into the structure of strigolactones, they have a basic nucleus or lactone nucleus consisting of three rings named as A, B and C. Now the ring A can be of different sizes. It can be a 5-membered ring, it can be a 6-membered ring, and there can be different substitutions on ring A and B. Now the ring C is attached through a enol ether bond to a ring D, which is an alpha beta unsaturated furanone moiety, also known as butanolide. Now the ring D is through enol ether linkage. This form an important character of this structure of the strigolactones, because ring D along with enol ether bridge is important for the biological effects that are exhibited by the strigolactones. And depending upon, or in simple words, you can say that ring D along with enol ether linkage is important or is the major biologically active part of the strigolactones. And depending upon the joining or the bonding between conjugation of ring A and B, these strigolactones can be classified into strigol type strigolactones or bancol type strigolactones. Now when either of the three rings present in the nucleus, either A, B or C, if any of the ring is missing, then we call them as another class of strigolactones, that is non-canonical strigolactones. But amongst all these three classes, the presence of enol ether along with D ring is a must to exhibit the biological effect of strigolactones. So here you can see different types of or classes of strigolactones. First one is the strigol type, wherein you can see the conjugation of A and B and C, and the second one is the bancol type, and third category is of non-canonical type of strigolactones, wherein you can see that ring B and C are absent. It has an open nucleus. Now we straightway move on to the signaling mechanism of strigolactones, and the major components that play an important role in the signaling system are the receptors of strigolactones. Now, various reports have shown that these receptors are mainly present in the roots of the plant, and different receptors identified in different plants have been given different names, like dwarf 14 or D14. It was found in rice, and this was the first receptor that was identified. Then ATD14 when the receptor is present in Arabidopsis, or DAD2 receptors when they are present in Petunia plant. SCF complex, as we have discussed already in the case of jasmonate, SCF complex is also present in the strigolactone signaling system, and we know that SCF complex is a mixture of three proteins: SKP1, culin, which is a ring box protein, and F box protein. And in case of strigolactone signaling system, MAX2 and D3 are the F box protein that form part of the SCF complex. And this SCF complex is nothing but a repressor protein complex which inhibits the signaling of strigolactones. Then we have the nuclear repressor proteins D53 or topless protein, TPL, again topless related proteins, TPR. We again have discussed that again these are the repressor proteins which bind to the transcription factor, thereby preventing the expression of the genes. Then different types of transcription factors also form the component, and the major transcription factors are teosnite branched 1 or cyclodia proliferating cell factor 1 family proteins, or the TCP proteins. So these are the transcription factors. Now the important thing about the strigolactone receptor, specifically D14, they have 4 helicals in their structure, as you can see in the picture. Now these 4 helicals are arranged in such a manner that they form a cavity. You can see that cavity in the figure B of this figure, B part of the figure, and in the magnified C part of the figure. So this moiety or this cavity, I will say, is important because this is the place where strigolactone molecule binds to this receptors, and whole molecule does not bind, only the ring D, butanolide ring, along with the enol ether linkage that fits into this cavity. And that is why I said that ring D along with enol ether linkage is essential for the activity of strigolactones. So that ring D fits into this cavity, thereby initiating the downstream signaling pathways. Any kind of modification in the cavity, which you can see in the D part of the figure, the cavity structure changes, and this prevents the binding of strigolactone onto the receptor, thereby making the plant insensitive towards the strigolactin effect. Now apart from binding of strigolactones, these receptors have enzymatic action also, as they causes the hydrolysis of the strigolactin receptor complex. And what is this hydrolyzing effect? We will discuss further ahead. So here you can see in this picture that the strigolactone molecule binds into the cavity of the receptor, that is D14. And once the strigolactone molecule binds to D14, its enzymatic action activates, thereby causing the hydrolysis of the strigolactin molecule, which causes the breakdown of the enol ether bond, making ring A, B, C free from the complex. Now this receptor is having only the enol ether linkage and the ring D of strigolactone, and once this complex is formed, it changes its structural configuration and become more or produce more affinity for the SCF complex, and this SCF complex is bind to the D14 strigolactone complex, which further causes the recruitment of the repressor proteins or the topless protein or TPR protein, that is topless related proteins, because these are the repressor proteins and they inhibit the transcription factors. So once this complex is formed, the transcription factors are free now. Now this complex or the SCF complex cause ubiquitination of the whole complex, which includes the SCF complex, the receptor along with the strigolactone, and the repressor TPL or the TPR proteins. And once this whole complex is ubiquitinated, it becomes more prone towards the proteasomal degradation, and it gets degraded by the proteasomal enzyme. And once this happens, transcription factors are already free, and they stimulate the genetic expression, or they stimulate the expression of the genes that are responsible to produce the effect of strigolactones. So this is the mechanism how strigolactone produces their effects in the plant. Now we will discuss what are the various effects that are produced by the strigolactones. So you can see on the right side, strigolactones inhibit the formation of the gravitropism of the shoots. So it does not allow the shoots to move towards the soil. So shoots are moving towards the upside only. Then it also inhibits the branching of the shoots. It also inhibits the formation of adventitious roots in the plants, and sometimes it also prevents the lateral root formation in the plant. Now this lateral root formation is largely the concentration dependent effect. At optimum concentration, sometimes it promotes the lateral root formation, but if the concentration is not optimum, it inhibits the formation of lateral roots. On the other side, you will see that strigolactone promotes the elongation of internodes. It promotes the senescence of the leaves. It also promotes the symbiotic effect along with the various fungus that are present in the soil. It promotes the elongation of the primary root, and if, as I told you, if the concentration is right, it also promotes the formation of the lateral roots. So these are the various effects that are exhibited by the strigolactones. Now we move on to the next and the last class of plant hormone, that is salicylic acid. And we know that salicylic acid was first obtained from the plant Salix alba, which has a rich history for its medicinal use. And if you look into the literature, you will find that the famous physicians of the yesteryears like Hippocrates, Dioscorides or Glen, they all have prescribed the powder of the bark of Salix alba plant for the treatment of different types of fever, inflammation or any kind of pain in the body. Because this formed the basis for the discovery of the salicylic acid. And if we talk about the modern history, then Edward Stone was the person, he was an English philosopher or natural philosopher who successfully conducted the clinical trials on the powder of that Salix alba bark, which is also known as willow plant or the willow bark. So he conducted clinical trials for the treatment of fever and inflammation, and those clinical trials were the successful trial or the first trial for the salicylic acid. Then Johann Andreas, who was a German pharmacologist, he isolated salicin, the first compound isolated from the willow bark, and this salicin was found to be a glycosidic compound that was present in the bark. Then Raphael Perea, who was an Italian chemist, he break down that salicin molecule into the sugar part and the A-glycan moiety or the non-sugar part, which he named as acidae salicylae, which is now known as salicylic acid. So you can say salicylic acid was for the first time identified in 1838. Then him and Kolbe, along with his co-workers in 1859, for the first time gave the process for the synthesis of salicylic acid. But it was until 1899 when Bayer's, the company who launched the drug aspirin, and it was Felix Hoffmann who gave the synthesis or the method for the synthesis of the aspirin. We know that aspirin is nothing but the acetyl derivative of salicylic acid, which is known as acetyl salicylic acid. So Hoffmann was the person who first synthesized acetyl salicylic acid, and Bayer was the company who launched this product by the name of aspirin, which we still use today. So aspirin was a blockbuster drug, and it was the molecule which increased the interest of scientific community in the plants to discover more and newer and effective drug molecules. Now it was in 1979 when the role of salicylic acid was first discovered, and it was found that salicylic acid increases the immunity of the plant against different type of pathogen attacks. And it was Raymond White proved the hypothesis when he exposed the leaves of a tobacco plant towards aspirin, and he found that the leaves become resistant towards the tobacco mosaic virus attack. So this was the first experiment which proved that salicylic acid provides the immunity to the plant. And it was only in early 1990s when salicylic acid was declared as a plant hormone. So we can say it's a very young plant hormone. Now what is salicylic acid? It's a simple phenolic compound having a very simple structure, and chemically it is known as 2-hydroxybenzoic acid. And this simple molecule is responsible for generation of certain endogenous signals that causes increased immunity inside the plant. Now how it is, biosynthesis of salicylic acid occurs through two major pathways: phenylalanine ammonia lyase pathway, which is also known as PAL pathway, or isochorismate pathway, which is known as IC pathway. Now it starts with the shikimic acid, which produces chorismic acid as an intermediate. Now this chorismic acid through ICS, that is isochorismate synthase enzyme, gets converted into isochorismate, and it eventually gets converted into salicylic acid. In the PAL pathway, this chorismic acid gets converted into perfenic acid, which leads to the production of phenylalanine. This phenylalanine further gets converted into cinnamic acid. Cinnamic acid gets converted into benzaldehyde or cumaric acid or orthocumaric acid, and both these intermediates eventually lead to the production of salicylic acid inside the plant. So this is how salicylic acid is synthesized through these two pathways. Now we move on to the signaling of salicylic acid. As like in the case of jasminic acid, salicylic acid is also stored in plant in different derivative forms. It can be methylated form, it can be glucosylated form, sulfonated form, or it can be in the conjugation with different amino acids. But all these are the inactive forms, and only once these derivatives are degraded, they get converted into salicylic acid, and then only the effects are produced. Now, it has been observed and reported that salicylic acid travels through the plant through the phloem, and various experiments have shown that the phloem sap contains large amount of salicylic acid, which leads to this hypothesis that it travels through the phloem and all its signaling molecules or its signaling pathway passes through the phloem. Now, the signaling mechanism of salicylic acid can be broadly categorized into upstream and downstream signaling. In the upstream signaling, the major component are the two R proteins: tall interleukin 1 receptor nucleotide binding site, leucine rich repeat, which is also known as TIR, NBS, LLR, and this protein further interact with enhanced disease susceptibility 1, EDS1, along with its interacting partner phytoalexin deficient 4 or the PAD4. Now, once this TIR, NBS, LLR gets activated, it stimulates the EDS1 and PAD4 complex formation, and once this complex is formed, it positively regulates the salicylic acid effect. It causes increased accumulation of salicylic acid in the plant. The second protein is the coiled coil nucleotide binding site leucine rich repeat, that is CC-NBS-LRR. Now, this protein further stimulates non-specific disease resistant 1 protein, that is NDR1. The first protein acted through EDS pathway, this acts through the NDR1 proteins. Now, these NDR1 proteins, they further interact or stimulate the RPM1 proteins, RPM1 interactin 4 proteins, which are known as RIN4 proteins. And once this RIN4 proteins are activated, they enhance the pathogen triggered immunity in the plants. Pathogen molecules, or I will say the PAM triggered immunity, pathogen associated molecular pattern triggered immunity in the plants. Along with PTI, it also stimulates the effector trigger immunity in the plants. So, how it happens, we will discuss further in the lecture. The other upstream components are the nitric oxide or reactive oxygen species. Now, we have discussed that if a plant is under any kind of stress, whether biotic or abiotic, various kind of reactive oxygen species and reactive nitrogen species are produced inside the plant, and these species form a positive feedback loop that causes the initiation and accumulation of salicylic acid, or I will say they potentiate the effect of salicylic acid inside the plant, thereby providing the protection to the plant against those free radicals. MAP kinases is another component of the upstream signaling, and it has been found that different MAP kinases have different effect on the salicylic acid signaling. MAP kinase 3 and 6 have a positive effect on the salicylic acid signaling, whereas MAP kinase 4 has a negative effect on the salicylic acid signaling. Calcium calmodulin complex also affects the upstream signaling of salicylic acid. It has been found that binding of calcium calmodulin to the Arabidopsis transcription factor SR1 represses the expression of EDS1 and suppresses the salicylic acid's accumulation and the defense of the plant. Whereas if the calmodulin binds to PTI, that is PAMP triggered immunity induced calmodulin binding protein 60, it positively affects the salicylic acid accumulation and provides pathogen resistance in the plant. Now we discuss about the downstream signaling. So as we have discussed, NDR1 through the R protein, EDS1 through the R proteins are the upstream, major upstream signaling pathways of signaling acid, salicylic acid. Now the downstream signaling has the major component as non-expression of pathogenesis related genes 1 or the NPR1 pathway. Now the downstream pathway has two major mechanism. One is NPR dependent pathway and second one is the NPR independent pathway. Now in dependent pathway, TGA, CGT, CA cis element binding, or also known as TGA proteins, they further stimulate B-zip proteins or the basic leucine zipper proteins. Now these proteins are responsible for the stimulation of various transcription factors, and apart from the transcription factors, various other proteins, Nim interacting 1, 2, 3, which are also known as Nimin 1, 2, 3 proteins, and all these proteins collectively they stimulate the salicylic acid induced gene expression. Now the Nimin effect in the salicylic acid pathway has been found to be concentration or concentration dependent. If these proteins are overexpressed, they negatively regulate the salicylic acid, and if their expression is optimum or in low concentration, then they increase the expression of the salicylic acid induced genes, thereby increasing the pathogen related proteins, increases the expression of pathogenesis related genes, which increases the systematic acquired resistance of the plants. Now in the NPR independent pathway, it involves two type of transcription factors, WHY and WHIRLY, or the VIII transcription factors. Now these proteins directly bind to the DNA, single stranded DNA of the pathogen, thereby causing damage to the pathogenic DNA and hence providing the resistance or the protection to the plant against those pathogens. And various reports have shown that the NPR dependent as well as independent pathway, they work in tandem, they work synergistically to enhance the protective effect of the salicylic acid, or to stimulate or to further synergize the salicylic acid signaling pathway. Now once the salicylic acid transcription factors are stimulated, they further causes increased expression of different genes, and depending upon the time taken by the genes to express themselves, the salicylic acid induced genes can be categorized into two categories. One is the immediate or early genes, which basically exhibit their effect within 30 minutes of the salicylic acid induction or the exposure to the salicylic acid to the plant. Within 30 minutes they will start their expression and exhibit their effect. And the common examples are glutathione S transferase 6 gene and immediate early induced glucosyl transferase gene. Whereas the late induced genes, as their name indicate, they stimulate the expression of these genes occur at a later period in comparison to the early genes. And the most common example of the late induced gene is the SA marker PR1 gene. Now it has been found that salicylic acid signal, whenever the salicylic acid signaling is induced, it antagonizes the action of various other hormones like jasmonic acid, abscisic acid, or auxins. Whereas if jasmonic acid or jasmonates or abscisic acid are activated, they in turn also inhibit the salicylic acid effect. Now this is the diagrammatic representation of its signaling pathway. Now on the left side you will see there are salicylic acid binding proteins, or you can say these are nothing but the receptors. So in the upstream signaling pathway we will see that the two R proteins, they initiate the accumulation of salicylic acid. Now this salicylic acid one is produced, it inhibits the salicylic acid binding protein. Now this salicylic acid binding protein is responsible for the degradation of methyl salicylate into salicylic acid. So this is a kind of positive feedback mechanism. So if salicylic acid concentration is high, they will inhibit this protein, thereby causing accumulation of methyl salicylate in the plant. But if there is requirement of salicylic acid, then these proteins get activated and they cause degradation of methyl salicylate into free salicylic acid, which produces the systematic acquired resistant in the plant. In the downstream signals you will see NPR1 proteins, and NPR1 proteins largely occur in the form of oligomers. When salicylic acid accumulation increases, it further stimulates the intermediary proteins, which are TX family proteins, which causes breakdown of these NPR1 oligomers, because NPR1 oligomers cannot enter into the nucleus in their oligomeric form from the cytoplasm. So once these oligomers are broken down, monomers are produced. These monomers through nuclear pore, they enter into the nuclear region, wherein they further activate various transcriptional factors and that lead to the expression of the protective genes. So this is the NPR1 dependent pathway, and in the same diagram you will see salicylic acid also directly activates VIII and VIII proteins, which forms the part of the NPR independent pathway, thereby increasing the expression of the protective genes in the plant. So this is basically how salicylic acid exhibit its effect in the plant system. Now we will discuss about the effects of the salicylic acid in the plant. It has been observed that salicylic acid or the effects of salicylic acid are largely concentration dependent. Higher the concentration sometimes lead to derogatory effect. So the optimum concentration of salicylic acid should be there. Now salicylic acid has been found to develop, that is, PAM triggered immunity or effector triggered immunity or systematic acquired resistance in the plant against various pathogen and oxidative stress. Under abiotic stress condition such as high salinity or temperature, lower concentration of salicylic acid has been found to increase the germination rate as well as seed vigor. So this is an example how the concentration of salicylic acid will impact or will produce its positive effect. Optimum concentration of salicylic acid has been found to improve photosynthesis. They causes regulation of stomatal closure, thereby reducing the respiration and improving the immunity. They also lead to the chlorophyll and carotenoid content in the plant, which also protect against reactive oxygen species, and it improves the activity of photosynthetic enzymes also. They have also been found to improve the vegetative and root growth in the plants by enhanced cell division and the better leaf growth, whereas higher concentration of salicylic acid has been found to cause senescence in the leaf, and it has also been found to induce the flowering in the plants. Thermogenicity is an important effect of salicylic acid, and it is the genesis of the heat in the plant. It was Jean-Baptiste Lamarck in 1778 who for the first time reported this effect in the arum lily, and he found that whenever flowering is there, the temperature of the male reproductive organ was increased tremendously in the flower. Later in 1937, Van Herk isolated a group of compound named as calorigen, and he stated that calorigen is responsible for the heating effect of the lily plants when he was working on the voodoo lily. Later, Raskin in 1987, he prepared extract from the spadix of voodoo lily, and on analysis of that extract, he found that it mainly contains 70% salicylic acid, and this is how it was discovered that salicylic acid is the molecule that was responsible for the thermogenicity of the plants. And it has been found that specifically in case of voodoo lily, during the flowering conditions, the temperature of the spadix increases almost 15 times more than the surrounding temperature. So these are the various effects that are exhibited by the salicylic acid. Largely it enhances the immune response through pathogen triggered immunity or the PAMP triggered immunity, effector triggered immunity, or the systematic acquired resistance. It also protects against stress conditions. It has a thermogenetic effect. It regulates the closure and opening of the stomata. It modulates the nodulation on the plants. It regulates the growth and development of the plant. It also regulates the germination of the seeds, causes leaf senescence, has an effect on the yield of the fruits. It also affects the respiration of the plant, thereby also, as it regulates the opening and closing of stomata, it also regulates the respiration, and it prevents or give the plant enough energy, or I will say enough support to lead to the repair of the damaged DNA. Vis-a-vis, it also causes damage of the pathogenic DNA, thereby protecting the plant. So friends, this was all about strigolactones and salicylic acid. Thank you.